nutrition-rich rice, and also engage in improving nutritional quality of soybean protein and using seeds as a bioreactor for high-value phytoplankton protein production. And I think I'm sure you'll be very interested in know how Professor Sun, during his long, career in scientific research, and we're glad to have Professor today to share with his many years of research how he keep motivated and how he his experience in this talk today in science events, the Springhead of motivation and innovation. And without further ado, let us have a round of applause.
Thank you, Professor Wang, for the introduction. Today I am going to share with you two parts. First, summarize my previous 40 years, actually 42 years research. And then from there, I would like to talk about the motivation and innovation, the Springhead of motivation and innovation. I think it is particularly important for our young scientists to share with you my experience, my learning from those innovation books and etc. I explored in the past few years. Okay. First, so this is the lecture series to attribute to Professor Yang. So I will give three talks because the level of the audience is different. So today mostly graduate students, professors, and then Friday will be a diverse background of undergrad, and then Saturday will be the general public. So I will talk differently. So today will be mostly more scientific. And I graduated, as Katie said, 1966 from New Asia College at the old campus in Hokkaido. And the chairman was Professor Yang. And the year we graduated, we have 15 students, and 8 female, 7 male. And then usually we have a graduate farewell dinner. The student invite professors. I think nowadays we don't have this thing anymore. Maybe we have to invite the graduates to dinner. Okay. There are many memories about Professor Yang, but I just want to mention two. So Professor Yang was the first one to discover this gold-fronted reactor bird in Guangxi province. So in 1932, 10 years before I was born. And so the name in Chinese his name is Yang Gongwei, so it's called Gongwei, right? So it's a very pretty bird. It's a songbird. And the front is a golden color. That's why I call gold-fronted reactor. And another thing I remember Professor Yang very well is that he really pushed us to go abroad to continue the study. So under his influence, 15 of us, 14 went abroad to do a master or PhD degree, and only one girl stayed in Hong Kong, but subsequently she also moved to foreign country, although not studying. So these are two things I remember. But there are many other things, maybe Saturday I will talk about, Friday. Okay, so then move on to my previous, some 40 years research path, and I'll very briefly summarize. So maybe you can see how I try to do my research and what motivates me and what are the things there maybe younger students can learn from. So I did my PhD at the University of Wisconsin-Madison. It's a very beautiful campus and with 4 lakes, and the area here is the university campus. So the red arrow pointed to my supervisor, Professor Timothy Paul. So last month he passed away at the age of 78. He's a very dedicated professor, and the picture we have is during Halloween. We all dress up for a party, and you can see here with a lot of hair, still black hairs. All right. So then go on to my research as postdoc. So I went to Wisconsin for my PhD in 1970 and got my PhD in 1974. And then I stayed on in Professor Tim Hall's lab as postdoc and then become research scientist. So I will brief you about my postdoc and research scientist work there. So during that time, Professor Hall was very young, junior faculty, very poor, not much money, not many instruments. So I have to work with the genetic department's Professor Oliver Smith in his laboratory. He was very generous and allowed me to work on the things I work on in his laboratory. But he has so many students and postdocs, so I can only work during night time and during weekend. So it's very hard work, partly because of the physical constraint. So of course, you know Oliver Smith. Later, he got the Nobel Prize, gene targeting. So he was one of those. And remember, he told me to be a good scientist, you need to have 3 things. 1, a passion for science. 2nd, you used to have a hobby. So his hobby was flying airplanes. And one time, he came to my house and he taught my son how to fold the paper airplane. And indeed, he can do it, fly very, very far away, very impressive. And 3rd is he used to have a good family. So he then he married his postdoc later, a young Japanese scientist. All right, so I did my work. He taught me quite a bit of things. So then my work is, during that time, we have animal gene clones. We know animal gene have intron, etc. So we try very hard to clone a plant gene, but no success. So then there's a saying, the plant gene is non-clonable. So then we decide to clone it and pick a system called French bean. It's the green bean you can buy. Why we work on a green bean? Because as you can see, my work during development, we take the beans from very young to mature. You can see some protein come out very strong. Almost 50% is this kind of protein we call globulin 1. So 3 subunits, very similar. So it is very abundant protein. So they express very strongly with a lot of messenger. At that time, we thought maybe we can clone it. That's how we want to do it. So then the problem is at that time it's very hard to make extract good plant DNA, as you know, right? We have cellulose, we grind very hard. Actually, then you all you got is very broken DNA, and also the plant genome usually big, and the library system. We use phage library system. It's not efficient, so it takes a lot of time to get DNA, to clone, to represent several times of the genome. So then you can totally cover the gene. So it becomes very, very difficult. On the other hand, I also need to borrow somebody else's place to work. So we're very, very challenging. So at the end, of course, we did it, and we cloned the physiology gene and did sequencing. And remember those days we do sequencing, you have to use maximum Gilbert. You have to pour your gel, right? And cut the DNA and label each fragment with E32, and then you run the gel, put on the X-ray film, and exposure, and then you see the band. And then you use that as a guide to go back to the gel and cut the piece of gel out and extract the DNA, and then do the sequencing, one piece by piece, and make it up. That's why it takes so long. But anyway, we clone this gene and find out this phase 1 gene also contains 7 introns. So that, of course, is the first time we discovered plant gene also contain intron. And also, of course, you need to clone the plant gene, then become possible to look at the sequence. So that was my work during the postdoc period. There are some other works I will not go into it. Okay, so then I complete this publication in Nature in 1980, and then at that time many people come to recruit me, and I was in Wisconsin, very cold, from November until April. It's totally frozen. I died to go to California, so I decided to go to California, and it's Arco, the oil company, the number 7 biggest oil company in USA. The boss owned the most land in United States. So I went there. The salary was at least double, and everything is very fine. But I really enjoy working there because we recruit 50 PhD and professors to come together. And during that time, the boss of the Arco said the oil will run out in 50 years. The next biggest thing will be to feed the people. So we do biotech, improve the food system. So then he put in a lot of money, 50 PhDs. They have no idea what we want to work on. So we can do everything we want to do. Publication, do everything. So we were so happy. You don't need to apply grant, you don't need to teach, you just work. And you hire postdoc, etc. So I have a very good time. So the people here is my group, and I stayed there, work on several projects, but I will just mention 1 or 2. So, so this will be my research at the institute. So it's an industry, research institute. Okay. So I was a senior scientist, and then principal scientist, and then director of molecular biology. I stayed there 7 years. Professor Wong said everything is 6. Actually, in the US, average is 8 years to 9 years. People will move from time from one job to others. You can see mine is about the same. The longest time is in Chinese New Year. I love the place. Okay, so then here I try to improve the nutritional quality of the soybean and other cereals. That was my interest. Okay, very briefly, there's about 8 amino acids is essential for human being and the one-stomach animals because they cannot synthesize and also resynthesize. So we have to intake from outside. These 8 have to be different animals, we have to be different proportion. So so-called essential amino acid, we have to have these 8 amino acids at the same time and also the right proportion. Then our synthesis system can make all kinds of proteins, hormones, antibodies, etc. So it's very important. If one amino acid becomes deficient, the rest will be metabolized and not used for making complements of protein. Okay, so then because it's an industry, right, we have to think about down the road maybe we can apply it. So I look at cereals, all deficient in lysine, all the beans are deficient in methionine, and the roots and tubers, it's pretty much contribute to our plant food. The others will be both methionine and cysteine. So the thing is how to raise the percentage of this. And they calculate, just in the US alone, they use soybean squeeze the oil out and the protein, about 40% dry weight, they will use to feed animals, to feed chicken, to feed pigs. So just every year in the US alone, because you use soybean, you don't have enough refining. You use corn, you don't have enough lysine. So you have to add the artificial synthesized refining and lysine. Just each year in the US, you spend $120 million to supplement the animal feed. Lysine will be $70 million. So it's a very, very big opportunity to the industry. So I work on this part. And then I start thinking, how can we improve the beans refining content? There are certain ways, right? So I search, I study, and then from a Brazilian PhD thesis, I find out there's a protein in Brazil nut contains 18% refining. Soybean only 1%, so this contains 18%. So the idea is, this is 40, 50 years ago, this slide. So the idea is if we can isolate this refining rich protein and get the gene and put it into soybean or any legume and let it express, and then we will have this protein very rich in refining, then we can raise the beans refining content. So that's the idea. So then we have to start doing the project. Turn out, so I study the whole United States, where I can get the Brazil nut. You can buy it from the grocery store during Christmas especially, but they are heat treated, so the DNA is inactivated. So you have to go to the original place. I look over everywhere, 2 trees in Hawaii, all the others are in the tropical jungle. So we have to go down to the tropical forest in South America. So this is a picture in 1984. Right? Many young students still know. Yeah. Some molecule somewhere. Anyway, so you go down, you can see the Amazon. You can see the river. This is my that time. So I ventured into the town close to the jungle area, and the jungle is so huge, so you cannot just get into it. You get lost, right? You don't see the sky, you don't see the road. So we have to hire some guy and lead us into. Really, we have put on raincoat so wet. So that was the time I was totally eye-open. So many different plant species. At least I don't know. So many insects, so many small animals. So I become fall in love with these tropical plants and animals. Anyway, into it. So then you find some Brazil nut trees, so tall, 200 feet tall, and there's no branch at the bottom. So how can you get up to get the fruit, right? You can see some fruit here. There's no way you can get up, right? And so this is very tough. You see, some are 200 years old, this 12-year-old is already very tall. So then, when you get into the jungle, it's 200 feet tall, and also very old, you cannot touch the bottom 4 feet. They're so high up. Because in the tropical jungle, you have to compete for the sunlight, so everything goes up, right, to compete. So then, all you can get is the Brazil nut fruit become mature and rotten and fell on the floor, the base, then you can pick up. Mostly the nut are not workable. So I have an idea, maybe I can train the monkey to go up to pick one. So this is the guy, he has a monkey. So I feed the monkey with Coca-Cola, so he likes it. So I bring him to the tropical jungle and let him taste the Brazil nut. Maybe next time he can do that for me. But after coming back, luckily, we find out in some open space, right? So people grow Brazil nut. You can see in the open space, Brazil nut tree has branches. Okay, so we find one tree with another lower one. So then you can hire some local boys, climb up here, use a ladder to go up, and use a very long pole to get a fruit very, very far away. So from there, we can collect the fruit and come back, cut it open, and this is Brazil nut fruit. Tastes very good, fresh one, not the one you got from the grocery store. Then I do some preliminary study. Is truly, if I can engineer this gene into the target plant, would that increase the methionine content, right? So I did some feeding experiment. So I feed Japanese quail with Brazil nut protein, right? Sorry, with the soybean protein. So soybean doesn't have enough methionine. So after 16 days, Japanese quail grow very poorly, right? They need methionine to grow, right? It's essential amino acid. And also our hair and feather needs methionine to synthesize. So we can see a deficient in methionine, the feather grows very poorly. If I feed the soybean protein, I add some Brazil nut extract or protein to it. You can see the same area grow very well. So that means if I can indeed put this gene into the plants and seeds, express the effect should be there. So then I start cloning the gene, and indeed you can see the sequence, so many methionines, very, very rich. So then we put it into tobacco plants to test. Indeed, it express and raise the methionine content to a significant level. So the Science News attributed this to the first time using biotech, you can improve the essential amino acid of the plant. So that was the work. All right, so far so good, right? So all the industry is so exciting, right? So much money there. So they came in and they bought the gene for $6 million US. Okay, it's $6 million. I said, wait, you have to do some tests, right? Is it a good protein? Is it safe? They said, no, we will take care of it. So it was sold for $6 million. All right, so they put it in the soybean, so excited, want to go to onto the market, but you have to go through safety tests, right? It turned out somebody, if you eat this engineered soybean, you get allergy. Okay, because any protein actually, there's some population, 1% will get allergy to it. But once you have these problems, the company stop it. They cannot afford to release it. So the whole project stopped. So I said, see, I told you, it's a more money. I will find out. But anyway, so they stopped the project. So now the Greenpeace people still use this case as Brazil nut engineered soybean will harm people. But the thing is never went onto the market, okay? Remember this, never stop. The company spent so much money to import, right? Pioneer, they cannot afford to harm one people. But anyway, so my big dream didn't work, scientifically work. But on the other hand, the news, okay, San Francisco Chronicle, New York Times, wow, the nightmare of different genes means genetic engineering plant can cause allergy, right? So it turned out to be a good thing. Okay. So now we test all the GM food very extensively on the allergenicity before you can release it. If you don't find out these, right, that's not too good. So 有時好心都做好事嘅 right? So that's what this particular project, okay? All right, so at that time, it's about time for me to move, about 6 or 7 years, right? According to American law. So I at that time, the oil company's business not too good. So the institute asked us to go out to look for projects and see what they want to do. So I feel it's no freedom anymore for me to do anything. So I was so fond. I love the tropical journalism. So I look for the 2 reasons I want to move to tropical region. One, I have severe allergy in Wisconsin and in California. So I want to move to a place without allergy. So only 2 places in the US, in Florida and Hawaii, there's no allergy. So I thought Hawaii is better, right? Closer to Hong Kong. So I moved to Hawaii. So indeed, just in the campus, you go to Hawaii, you can see all these tropical fruits. I was very excited. So during that period, I cloned some 12 genes, very different genes from tropical germplasm. Because my idea is, in the future, with the rainforest going, so many unknown genes may be very useful in the gone. If we can use molecular technique to preserve all these species, maybe not with tree, we can make library, we can store it for a long time, and then we can code still to isolate the genes. So I work on tropical genes. I isolate many tropical genes. I will just pick 1 or 2 examples. So I find out from a Chinese medicinal journal, I find out there is Chinese herb. It's very sweet. So turn out inside there's a protein 400 times sweeter than sucrose. So the idea is maybe I can extract the protein and put it into something that will be sweet but with no calorie. Be good for the diabetic people. So I went to Kunming and collaborate and we clone the gene. So I collaborate with China's Vegetable Center, because in China people eat watermelon a lot, but the diabetic people cannot eat it. So if we can find a watermelon not sweet and put this protein in, bring sweet, that would be good. So that's the project I give them to work on. Another thing is I find out a tropical bean called wing bean. I find out now you can buy it from Park and Say Buckeye. So this wing bean, so I identify a gene contains very high lysine. This gives me opportunity to improve the cereal, which is deficient in lysine. And finally, I also engineer tomato to be resistant to virus. I also help Hawaii scientists to set up molecular programs to generate papaya resistant to ring spot virus. We buy the Hawaii papaya, now mostly virus engineered. So that was my work at Hawaii. And then in 1996, again, so I returned to Hong Kong, and I work on some other things. So what should I work on? So I look at 50% of the world population and 50% Chinese. We use rice as staple food, right? So that's important. So then, as I mentioned, all the cereals deficient in lysine, so we need to improve about 40%. And there are literatures. If you don't have, children have problems with protein balance and etc. and eat poor proteins, if you give them high lysine corn, you can alleviate the problem. So we feed some feedings with rice and add some artificial lysine to it. Indeed, with increasing percentage, you can see the growth improve, right? So that's the preliminary data. So then you can see the wild type growth, eat the wild type rice, and then with increasing addition of lysine, you can see promote the growth. Okay, so I will not go into detail. So we have 3 different approaches to try to improve the lysine content. One is improve the free lysine content. One is to express rice own lysine-rich protein, and another one express a foreign gene into it. So we all raise very significant lysine content. So we have field tests many times of this high lysine protein, and you can see eating the high lysine, the animal grow much better than the regular rice. And for the girls, don't afraid. You eat the rice doesn't mean you will very big and fat. Actually, you know, in normal diet, 2 times bigger than this one. It just improves your normal growth rate. So then, this particular lysine-rich rice, we have subject to the China agriculture food, GM food safety test. After all the tests come back, the report from different tests, allergy, everything, so be safe. So this is to the stage that this year we will do some environmental tests. If that goes well, maybe we can get the farmer to try on it. So I'm very happy, finally, something I can get close to the end product. So my other postdoc, Eric, also, because the children just eat rice, very skinny, very stunning, right? So we did some tests. The high lysine rice indeed improve their bone development and also the muscle development. So these are something we are working on. And rice, besides deficient in lysine, it also have no vitamin A, very low vitamin E, not sufficient iron and zinc. So that's the other deficiency of rice. So as you know, Patricus and Peter Bayer, right, in Europe, they already engineer rice with the beta carotene. You see, all the plants only can contain beta carotene, no vitamin A, right? This is the precursor. When we eat the beta carotene, our body will digest it, split into 2 molecules of vitamin A. So they engineer the rice to have beta carotene, right? So this is the regular rice. This is the first generation of golden rice, pale yellow. And then the second generation contains 32 micrograms per gram of rice endosperm. So you eat about 200 grams of rice a day. The children get enough for it. So I wait for about 2 bowls of rice. So this the golden rice already there. The thing is, it's still deficient in lysine, deficient in iron, vitamin E, etc. So that's why Bill Gates Foundation and we support these grand challenges. So there are many projects, and we think that we will support the plant area to create a nutrient-rich, stable plant for the poor people. So they pick banana, right? Africa, they eat banana, cassava, and also sorghum. One is the rice, to engineer a rice with high beta carotene, vitamin E, good protein, and enhanced iron and zinc. So the idea is use the golden rice already have vitamin A, provitamin A, and to engineer into it. So they will have, the rice will have good protein, good vitamin, and good mineral. We call this provitamin rice. So we form a consortium with scientists from different countries to tackle on different problems. So I'm responsible for the good protein, the lysine part. Okay, so we always have meetings in Africa to feel the need of improve crops, and the aim is to generate natural label, genetically engineered label, golden rice with vitamin A, vitamin E, iron, zinc, with lysine. So that was the thing we tried to do, and I complete my project, lysine-rich rice. So you see, we have 7 different scientists. We don't put all the genes into one species, so farmer can grow with everything there. We individually do the thing. So I do lysine, produce rice with high lysine, the other with high vitamin A, vitamin E. So then we will test safely and then give to the farmer. Depends on what they want. They want vitamin A and lysine, they will cause it, right? So we don't need to force them to have everything. Maybe the rice in Vietnamese have enough iron, right? We don't need to have that. So this is how we decide to do. So I complete my part. All right. Another thing would be, so I improve the lysine content of the rice. Another thing will be trying to improve the yield. That's very important. For yield, so I find out hybrid rice was invented by China, by Professor Yuan Longping. It is high rising as of the hybrid rice, at least 20% yield increase over the best variety. So right now, half of the fields growing hybrid rice, so we increase enough to feed 100 million more Chinese people. So I thought if I can help improve the yield, that's why we collaborate with Professor Morris Koo, Professor Yuan Longping. We try to move the corn's photosynthetic gene because C4. I have no time to go in. It's very efficient in capture sunlight. So we try to move it into the C4 rice, generate C4 rice. So we have generated this rice and we test in the field. Indeed, the C4 gene express increase the activity and etc. The yield increased from 1 to 7%, and which is quite good because right now the rice every year average over the whole world is increased 1 to 2%. So if we can increase 1%, that's good enough. But of course, we agree we want to increase more. But the problem is the seed is not totally filled up. So there are empty seeds. So then we finally decide why the engineer, the photosynthetic gene is not filling up all the grains. We analyze because the promoter views may be not the best suitable one. Because we find out the gene expressed in the mid period, they will turn off. So that's why at the end there's nothing transport with the seed. That's what we thought. So then we try to improve that. And also, once you harvest the seed, the rice, there are many starch still left in the stalk, in the leaf. So maybe we can mobilize those also transport into the rice. We can increase the yield. So we engineer 12 contract contain 20 genes, mobilize the starch from the stalk and also the leaf to move into the seed, and also enhance the expression of the C4 gene constantly until the seed set. So we already get the plant. So then the next thing will be we will test this in the field in Hainan, in Changsha. So that is the part of the yield increase. So then I would like to summarize. So in my first 5 or 7 years, I pretty much work on basic research. All right, try to understand the gene expression and structure and regulations. That's my phasing work. It's very most challenging, partly because the technical problem at that time. So my second phase is working at industry. So that was pretty much applied research, my pursuit work. It was very fun and unexpected, right? The third phase in Manoa, Hawaii, I pretty much work on move to apply basic research. I become more interested in doing something quicker to benefit the community. So I do some basic research, understand, and then I apply it. So if I come back to Chinese school, I pretty much doing the same thing. So this is the, I said now I'm very excited about the work because I'm going to the end, almost have a rice close to be able to release. You see Monsanto, those spend billions of dollars to get a product onto it. For me, I'm single. I'm fighting for so many years to get something onto the end, right? I don't have hundreds of people working on it, so I'm very excited I can get to this page. All right, so I want to acknowledge so many advisors, friends, colleagues, and then I want to also acknowledge my students, and also I frequently go to the rice field, to the village. I listen to them. What is their problems? What is their needs? What is their dreams? I think this is very important to motivate the PhD to work on the things that I'm working on. So now I have maybe 10 minutes. I will go to my second part. So then for the young students, or postdocs, or graduate students, so do you ever afraid of being a scientist? Do you worry about being a scientist sometime, right? It's unknown, right? And I get a job, that's my laboratories. So I think very essential. You should find out two things will help you. One is your motivation. Second is innovation, right? So because you have motivation, you will keep going on, right? If you have innovation, science, after all, is new ideas, new approaches, breakthroughs. Where are these from? It's all from innovation, right? So if you can take care of these two things, I think you will feel better to be a scientist. Okay, so I become very interested in these things. Professor Kim Liu, our previous Vice Chancellor, he's very good in calligraphy. So he gave me this particular piece of his work. It's called Chinese 還佢打得清佢唯有後水浪. Roughly, tell me why spring water is so clean. Because it is running water coming from the Springhead, right? So looking at the whole point, actually, is Zhu Xi is a scholar in Song Dynasty. So he has a study place next to a half an acre of pond. So he looked outside, the pond water is so clear. The sky, the blue sky, the cloud can all see clearly. So that's why he asked, why the water is so clear, right? Because there is a spring water coming out. So if we know the Springhead, we know the origin, where is your motivation from? Where is your innovation from? Maybe we can solve our problem. So if you look at what motivates scientists, okay, curious to see how nature works, very natural, right? How nature works. Second, inspired by great discovery, right? Right? You have exciting, right? You like to know the unknown, right? It's very challenging. Things unknown we want to find out, right? And also, you're a useful person. Do something useful, right? These are very natural starting. And some people just like to do something in the lab, right? Trying this, trying that, right? That's another things you want to be a scientist. Another group of possibility is you want to serve the humanity. You want to help the people through science, right? And some people want to have reputations and power. I will explain further, right? And of course, some people want to take this as a job to earn a living and even make a profit, right? So these are all the different kinds of motivation. So maybe you should think about what is your motivation, right? Once you find it, I think that's the point. So in sum, purely individual satisfy your intellectual curiosity, right? That's one kind. I would say probably trying to understand, trying to connect things, trying to explain things. This is more kind of basic research. Of course, not totally, right? kind of, you satisfy your self-curiosity. Another group is to improve the scientific community. This can be more complicated. For example, you do research, you publish, you want to share with others, right? And your discovery was recognized as a scientific community. You give credits. People fly you around to dine and wine you and give lectures, right? So these are credit given, recognize you. You publish, also recognize you. They cite you, citation. This is all something, right? Excite you. You're doing good work. People say this is a good scientist, great scientist. Right? Give you honorary degree, give you an award, give you an academic mission, right? This is also reason, right? And also invite you to give talk at the conference, panelist, speaker, right? All these are common to this category, right? Some people interested in this. And of course, another group is to improve the humanity, right? So hopefully your thing can be applied, people can benefit, can use. I would say kind of more close to applied research. But it's not this simple, right? Everybody, not just purely this and purely that. Actually, maybe it's a mixture. It's very hard to tell, right? So could be many things mixed together. So I encourage the young students, graduates, really sit down to think about what motivates you. Doesn't matter what motivates you. Once you find it, you will have this source of energy, motivation to push you through, right? There are so many obstacles. Things are very difficult to discover now. So you should know your springhead of your motivation. The other thing will be innovation. So innovation is the creativity, right? Your creativity, where it comes from? It's come from your imagination, right? Your imagination, you need to have some knowledge to imagine. Otherwise, things are not working. And once you have creativity, you think about something, you put it into use to improve, to come up with new idea, to make some change. That will be innovation. And further down will be entrepreneurship. You make it into a business. So the relationship is like this. So everything is coming, I think, from your imagination and feeling. That's why Einstein said, imagination is more important than knowledge. Why? Knowledge is limited. Imagination encircles the world. That means imagination can bring you everywhere. Right? Knowledge may not. Imagination can bring you anywhere. That's why imagination is very important. But where is the imagination coming from? The brain is a creative machine. Used to be, we believe creativity is a divine thing. It's coming from somewhere, right? But people now believe it's within us. Okay? Everybody have this imagination, have innovative ideas because it's here, right? So we have 100 billion neurons. They organize, they analyze things every second, right? So then give you the best choice, best solution. When you have difficulty, give you the best solution to meet, right? When you cross the street, you look at both sides. Estimate the car. You say far enough, I can cross now. It's calculated here. Making all kind of decisions there. So in fact, it's here. So our springhead is your mind. Okay? So everybody have this innovative potential there. The thing is how to use it, right? So everyone have. So I have many these pictures. I show you. Everyone have innovations, right? You can see it, right? You can make a seat easily, right? You can sit on a bottle, right? You can drink the things the teacher doesn't know. Anywhere you have this innovation, right? So I would say everyone have. The thing is just how to promote, how to enhance it. So I give you some example. I wish our students can do. First is a positive attitude, right? So when we have a problem, don't shy away, don't run away. Take the problem as an opportunity for you to innovate. Right? Then you will have a positive attitude. Every problem is an opportunity for you to overcome, for you to think about a way to solve it. Second, be a keen observer. Right? So if you are a keen observer, you can see something that others don't see. That's a good, right? See something others don't see. And also for the details, right? For the patterns, right? Look at the big pictures. Look at something inconsistent. From there, you can do something, right? And also find a detail into the very origin. For example, if this solid wood, right? Get into it to look at it, to the origins, right? And also develop your knowledge. That is the thing to fuel your imagination. So this is for clues for you to innovate, okay? So it's very critical skill, particularly for scientists, to be keen observer. For example, just one example I have made. So this cockroach, well, as I said, look at something, people don't see it very good. If you look at something, everybody see it, you can still think about something other people don't think about it. That's even better, a higher level, right? You saw cockroach, right? So then people use it, put on a tiny computer. So this Jiao Tong University student now can use their brain wave to direct the cockroach to go left, to go right. There's nothing connected, right? So this innovation, see the cockroach, then you can do something like that. Another thing is to reorganize, to reframe your problems, right? So that's why when you look at one thing, from left to right, top, front, back, and bottom, and inside out, then you will find many things. Once you understand, you can come up with new solution. If you look at problem, use your eye to see it. Use a children's eye to see it. Use a male, female, foreigner, right? Older people. Then you will have a different perspective. Give you a lot of chance to solve the problem. Fit for everybody, right? That's important. And also asking why. Always asking why, right? That's a very good example. So there's two small villages. Between is a river. So one day someone said, I come to you, you are a builder, right? I want to build a bridge to cross to the other village. Okay, you just do it. And maybe you should ask him, why you want to build a bridge? He said, I want to go to the other side. Right? So you ask why. Well, go to the other side, that's what it means. So you build a bridge. You can make a ferry, right? You can make a tunnel. You can do many different things. There is totally open. Then you can continue to ask why you want to go to the other side. I want to go there to work. I want to go there to see my girlfriend or boyfriend. Then totally many things will come up, right? You ask questions, open a lot of possibilities, and proofs for multiple solutions. Usually, something come up, you think about the first solution, usually it's very obvious. Force you fail to come up with second and the third one. Usually those will be more innovative. That's why we do brainstorming, so many people come together, right? And as Steve Jobs said, his idea is this creativity is not you creating something new. It's you link up all the previous products, experience together to make something new. Indeed, right? This iPhone, he didn't invent anything, right? He just put everything together, right? So it's already there. You just have to link it together. That's why our college encourage students go to summer intern, go to a cultural experience, go to visit. It's all accumulating the experience, and one day you will connect them together, right? So experience is very important. Another thing is to force different things to connect together, right? That's very important. For someone to go on an airplane, someone sitting next to you, talk to them, right? You learn a lot of different things, right? These are one of them. For example, myself, give you an example. I saw so many kids, right? Their book packs are so heavy, right? How can we do something? So that gives me the idea. I want to do something. That's my imagination to do it. So how to do it? I want to push the backpack with the leg. So that's my idea. You can walk alongside, right? That's my imagination. Right? That's what I have in mind. And turn out there's an Israel company already produce a luggage. It can go along with you, recognize you. Only you can open it, others cannot open. But I think this is not my final product. I want to help the student. You see, this cannot go on bus, right? Cannot go to the second floor, 50 steps. So I still have my dream to see if I can have that one. So that's some example to do that. Okay, another thing is to provide creative space for all these Google, everything, where their working environment is very stimulating, very relaxed. You see my grandkid in the nursery school, very relaxed. They just draw pictures, take them, so very free to encourage that we need to have that. And also, we need some pressure, because really, when we have deadlines, we will very well stimulate. Okay? All right, so finally, is that we need to accumulate knowledge with positive attitude, proactive, and also to practice the many things I have just mentioned. There are so many ways we can improve. So today, because time limited, I will just mention a few. You can practice every day. Hopefully, this will give you some boost to your innovation. So I have finished my first part. Thank you very much.
Thanks for his inspiring words. I guess time for two questions.
Okay, all set.
I can get maybe one question. You mentioned about Brazil nuts. It's actually quite interesting because in the sense that you look at some paper, you find this methionine rich protein, but there are actually many nuts around, right? But is it just an excuse that get you to the Brazil?
No.
Or is it any other system can do the same similar thing?
Right. So I want to improve methionine. So I search for methionine. I look at all the protein amino acid data and see what protein available with high methionine. And then I identify chemistry physics by chemistry student in Brazil. And the physicists analyze the protein. And actually in Brazil, they say you eat Brazil nut one day equivalent to a chicken egg. So that's why they already know about this. So then that's what they say. But I still isolate the protein, analyze composition, and later sequence. It's very easy to identify the whole line up in Brazil nut, and then you label it with methionine. When you run gel, this particular protein is just show up totally. Right? That's why you need to search, you need to learn the existing knowledge. Okay.
Yeah.
I'm interested in your analogy process.
Yeah.
Because it's something I actually do. A lot of CMOs like that. And even though you do a test, the effect will be delayed. It will not be something as because you express a foreign protein, it's a foreign material, it's plant, but you react to it. study the reaction of the host in this plant to foreign proteins or
So right now to pass this allergy test, first looking at the sequence, whether there are some existing allergy sequence there with the epitope we know, right? So we screen for 5 or 6 amino acids there, and if anything homologous is high, you have to stop there. Right? First, look at the existing data. Right? So if that pass, the next thing will be you can do some skin paper. Okay? The final another step is human feeding, but that have to be under the physicians. Okay? Can be very drastic things. Right? So, but turn out to be, I would say, any protein, there will be 1% of protein somehow allergy. It is sort of genetic things, so become very important. That's why subsequently, when I do some gene transfer, I always look at the internal gene now, identify genes already there. For example, the rice now, I look at the rice only, rice-rich protein. I just express high. So these are the things you need to do. It's not 100% guaranteed, but have to be very careful. So you also fully simulated digestion. So in the test tube, you put all kind of digestive tubes and put it in there. Things very easy to digest, hardly cause allergy. Right? You can't already digest all the detox thing on the body. And also look at whether they are resistant to heat. Usually, allergy protein resistant to heat. They very tough. Right? So we can also look at the heat stability. So there are many things simulated test, but the final test is the human. But that's very hard. So in my lysine one is already done, but that protein is from bacteria. So this is my first phase. Second phase, I use plant's own gene. Now I generate high lysine. That's how we try to do it.
When you cross 2 plants,
Yeah.
you generate new products. The plant that you see before. Right? So looking at the whole plant, very different. So there may be genes that rarely expressed in the seed, i.e. could be somewhere else. So we never seen it before. The plant system itself could be expressing new protein in the seed to counteract the immune stuff, new protein, new whatever in the seed. Therefore, it is itself is a plant protein or plant product, but it wasn't there before. And now it's there, and then the reaction. Even my wife has so many allergies now.
Yeah, I know. That's why she seldom come to deep. The thing is, if you are bringing genes together by crossing, that's already traditional one story. Okay? In molecular, we bring something is from different species, right? In faster, but it's not actually traditional one already crossed. Hybrid rice crossing wild species.
Yeah. Supernatural.
Yeah.
But then you're not doing things totally foreign
Yeah.
to the seed.
Agree. That's why the traditional breeding, you can cross rice with rice, you can go on the market right away. They don't ask questions. Actually, cause problem for like I said, right? It's happening. But in particular, genetic AGV1, because it's doing cross species, so we have to be very careful. That's all the one is looking at the technology.
Great. Okay.
Since innovation and imagination is so important to inspire people, do you have any favorite methods to do that?
People are feel nuts.
Actually, someone suggests, Professor, why you take so much time to engineer refining rich food, right? Can you engineer humans' gut? They can make refining, right? And then you can solve the problem, right? That's the ways. But how can I engineer you? But anyway, back to you. The way I'm talking about is some ways students daily they can try to enhance their imagination and innovation. But if I want to give you another example is, for example, in the New College, Dr. K.B. Wong and I work together, right? So innovation is the spirit what we want to develop in the student. So I cannot give them a lecture, force them to listen to me, right? So we have to do it subtly, right? 春風化雨, right? So what we are trying to do, many things like this, right? Just give you one example. Usually I go to college center Friday noon. We force hundreds of students sit together, right? And they are very noisy. They haven't seen each other so long, right? They see each other, they will talk, right? Right? It's very hard. I was invited to go to the course. So this is one thing. So in our college, we don't have this noon assembly now, just sit there and listen. So we have this high table dinner, right? We feed the student for 70 minutes. They eat. I think they talk enough, 1 hour. So then we come in, the speaker, they become very quiet. 就講食醉，講盡晒啦，係咪 ？So then they listen, right? And then we encourage you to raise questions. There are many examples. Just give you one. We dress them with gowns, so girls have to dress nicely. So opportunity to know the voice. So many good things, right? So they like it, and then they learn, right? I have been taught to them, respect the speaker, right? Respect yourself, right? I said in the ancient Chinese saying, 兼聽則明，你聽多啲你就會學到嘢，right? So we have to do something. So we do many things like this. We have a place encourage students to entrepreneurship. They can second-hand book. They can do something. We have many things we are doing. I'm knowing. Okay. Thank you. If you need to go, we go.
One question on that. Student from social science.
Sichuan.
Social science.
Oh, social science.
Actually, I learned from your slide that there are 3 steps from imagination to creativity and to innovation. I just wonder what is the difference between the creativity and the innovation? Is that the characteristics of science?
On Friday, because I will talk to undergrad, it's very different from here. It's much easier to give many examples. Actually, imagination, that's we are all trying to except one. Imagination is the idea, meaning 你想嗰啲嘢，你想嗰啲嘢，但係你夜晚發夢it's an idea, right? Sometimes when you dream about something, when you wake up, you forgot about it, right? So we always have some idea, and then how to release your idea become something more realistic to work out. That is the creativity, right? So creativity is the release of your imagination into something going down to a more concrete improvement, right? It's a second step. Second step. That's why everybody has imagination, right? That's what I say. 好似就像我看来，我看到很多I saw this book pad, so heavy, right? I want to do something. That's my idea, my imagination. I want to do something. And then, yeah, and then I want to solve the problem, right? So I force myself. Can I make the backpack in 2 legs? I force the 2 different things, and they work together, right? This is my goal. And then I want to work it out in my innovation. I need to come up with money. I want to design how the leg can go up, right? And then the further down, I want to make a business with the entrepreneurship, right? It's from here slowly goes up. Hope this clear. This otherwise that's my understanding. Maybe other people doesn't think like this. My logic is like this. Everything is here. You said everybody has imagination, right? I saw some example. So, right? Sitting on the Bible is something, right? So it's natural.
Okay, maybe the last question.
Hey, why you come here? From
It's
college.
a college student.
Okay.
From our college.
About your work, you mix different kind of rice together to get the metal one.
Rice.
Yeah, I think for millions of years, the natural method is most natural to have all these kind of rice to have them live there. So nature can actually protect them and protect nature itself. And now, because of human need, then you are creating some new kind of rice, maybe mixing with the others, so that human can use it better. Yeah, that's good. And for human, at least it's good, but what about for nature? I think so after so many years of changing, nature has already, I think it has already found a way to make itself to be protected as I think the best way. And now you are changing it, so are you kind of sacrificing the nature for the good of human?
Well, this is our own question at the time. It takes time to answer. I will just answer like this. I am part of nature. What I'm doing is also nature. Human being come to this stage, they are doing something. Okay? I am part of nature. Okay? You come to this stage of knowledge, you do something. It's natural. I'm natural. Right? So I just answer like this. Otherwise, it's very hard. Of course, you can let things go by. Right? Actually, the hybrid rice already everywhere, hybrid species all everywhere. We just do it in a way our technology now allow us. The thing is always risk and benefit. We have to decide on that.
In a sense, human civilization is a history of how human activity temper with the, with the nature. Although there's an argument on how there's a limit. That will be, think about this. That will be take some time to examine. But anyway, I think let's conclude this very inspiring talk with a big round of applause.
I want to do one advertisement. Any graduate student, postdoc, want to have a postdoc position at UC, San Diego Medical School, stay here. I can talk to you.
Okay.
UC Vegas, UC San Francisco, San Diego,